Hello, everyone, and welcome to the first MycoTalk edition of 2023. It's actually coinciding with another great event, the Fungal GRC, which is running in the moment in Texas. So thanks for so many of you who are joining. It's my pleasure to co-chair this with Olivier Kurzai. And we have two great speakers today, Malcolm Bromley and Andrew Ko. So the philosophy of the MycoTalks is high diversity, career stage, gender, geography, and field of research. It's very inclusive. And each of the speakers will have 3 minutes talk and 30 minutes talk, and it's a focus on different aspects of medical mycology. We will have Q&A following the talk, so please put your questions there, and we will follow on them. This is all recorded, so you can also see the talks after that on the YouTube channel. We have two upcoming talks in February and March. In February, we have Deborah Hogan and Chad Steele, and in March, it will be Julie Segre and John Perfect. So a lot of exciting speakers for this year. It will be great to attend. So there is also a similar initiative for trainees. So please register and attend. It's also a very exciting series of seminars here. So with that, I will, let me just stop sharing here. Okay, with that, I would like to introduce our first speaker, Andrew Ko. Who is an associate professor in pediatrics and microbiology and director of the cellular immunotherapeutic program in University of Texas Southwestern Medical Center. He received his bachelor's degree in biology from Harvard College, and he was then awarded the Rhodes Scholarship and studied English literature in Oxford University. Dr. Ko obtained his MD degree from Harvard Medical School, and he completed internship residency in the Children's Hospital in Boston. After which, and he has been affiliated with Dana-Farber Cancer Institute as well. After which he joined UT Southwestern. He has been very well published and very well funded investigator, and he has seminal contribution to our understanding of the process of colonization resistance of Candida in the gut, and developed number of studies and models where he added a lot to our knowledge of how gut disseminated fungal infections are happening. So with that, I don't want to take more time here, and Andy, the stage is yours.
Thank you, Ilya. Thank you for that very kind introduction, and thank you for the opportunity to speak here today. So let me this going in. Everyone can see the slides, I hope. So as Ilian mentioned, my background is I'm a pediatrician, and I'm a little unusual in that I'm duly trained in infectious disease and hematology oncology. And why I did that, is really primarily firstly interested in how cancer and stem cell transplant patients develop these often devastating bacterial and fungal infections. And so that has kind of been the kind of leading question in my lab for a long time. And before I start the obligatory kind of disclosure slide, I'm a consultant for this company and get sponsored research from Novartis and Prolactiv, and recently founded a startup company of my own, none of which should influence this talk today. So I'll give just a very brief overview of the things we study in my lab, and then talk about this concept of gastrointestinal colonization resistance, which we strongly believe is actually a very critical kind of defense mechanism by which the host prevents certain pathobionts from kind of gaining hold and expanding and causing disease. And then talk specifically about Candida, and then recently this kind of growing awareness among many microbiologists about the importance to study strain-specific differences as they may dictate phenotypes in the models that we're using. So Really, the longest ongoing project has been really trying to understand how bacteria and fungi can colonize the gut. What are the circumstances of both the pathogen and the host in which the pathobiont can expand? And then what are the circumstances that dictate whether when disseminated diseases can happen? And then in the last 6 or 7 years, we've expanded our focus to really understand how the microbiome itself modulates immune responses in general, adaptive. This graft versus host disease is an autoimmune complication in stem cell transplant patients, and more recently, how the microbiome influences responses to cancer immunotherapy. And that's really kind of transformed the lab, and most of the lab is really working on project 2 and 3, but obviously I'll be talking about project 1 here. So my whole journey into science really starts with this paper that I was made aware of. So up in the late 1960s, we finally were starting to give chemotherapy to patients, and it was having good effect with leukemia patients. And these were cytotoxic chemotherapeutic agents. So essentially what that means are chemicals that kill cells that divide quickly. And as you can imagine, cancer cells divide very quickly, but other cells in our body, the bone marrow, the gastrointestinal, skin, et cetera, can do it as well. And that's why you can also get these terrible adverse effects with cytotoxic or conventional chemotherapy. And what they found, which maybe seem obvious right now, is that a leukemia patient who's getting cytotoxic chemotherapy, those who developed fever and became profoundly neutropenic, so that's an absolute neutrophil count or ANC of less than 100. And just to put it in reference, you and I have neutrophil counts typically over 1,500, 2,000, so really profoundly neutropenic, that these patients would develop infections. And the #1 leading cause of infections at the time was gram-negative bacteria, particularly Pseudomonas aeruginosa. And with mortality cited as high as like 60 to 70%, just awful. Patients were dying of this infection, and everyone was really perplexed as to why this was happening. And so now this has become kind of standard of care to be really aware of those cancer and stem cell patients who develop fever and neutropenia. And it happens in about 80% of patients with leukemias. And I'd say it happens to about 95% of patients undergoing stem cell transplant. Now, strangely enough, of those, only 10 to 25% typically have a documented source of their BSI or bloodstream infection. So this has led to kind of what I call defensive medicine, in that we just give antibiotics almost all the time to prevent these infections. And the reason I even started doing science was this gentleman here, Philip Pizzo, who was my mentor. So Phil Pizzo was the head of pediatric cancer at the NCI, and then became department chair of pediatrics at Boston Children's when I was a resident and chief resident. And he took me under his wing and really got me interested in this whole concept of immunocompromised ID, which he kind of heralded in the late 70s and 80s. And this is one of the first seminal papers showing that in those patients that were persistently febrile and neutropenic, that you really needed to add antifungal therapy, because those patients were at highest risk for developing disseminated fungal infection, primarily Candida, right? And then a study in 1992, which is now 30 years later or earlier, was a kind of a definitive control trial showing that azoles, in this case fluconazole, was really good at preventing fungal infections, meaning start it right away, not before fever and neutropenia, as soon as they walk in the door, and you could reduce the incidence of infections. So the current paradigm in treating our patients is we just use antibiotics all the time, prophylactic, empiric. We start them obviously, and we change them when they're febrile and neutropenic. Antibacterial antibiotics must cover Pseudomonas aeruginosa, and on the fungal side, it's really about Candida albicans, or Candida species in general are the ones that we target. And they're oftentimes admitted for intravenous antibiotics even after the fever resolves, but if they're still neutropenic, even if they're well appearing, and they continue until count recovery, right? So Phil Pizzo was the one who really challenged me to start thinking about these questions. And so I picked up a pipette for the first time when I was a clinical fellow at the age of 30. I had never done any science or no interest in science. And what I kind of gravitated towards, because I think it was most kind of least scary to me, was working in animal models. So we've developed these animal models where we can take inbred strains of mice, and we can really do this in various strains, black 6s, BALBs, C3Hs, from the variety of different vendors, which is important because there's huge microbiome differences in these mice. And we find that by using antibiotics, specific antibiotics, we could really promote high colonization levels of pathobionts. And again, in my postdoc work, it was Pseudomonas. We've done this with E. coli and Enterococcus, and obviously we've done it now this with Candida albicans. And then by giving the additional insult of either cytotoxic chemotherapy or radiation therapy, we can induce bacterial and fungal sepsis. And this has kind of been our go-to model for the years as we've kind of studied these things. And what we found is that in both associative studies in humans and in mice, these were the kind of three key host immune factors: microbiota kind of balance or homeostasis, whether you had neutrophils, and the degree of kind of mucosal damage, or what we call in humans mucositis, and whether that would dictate whether you had a disseminated infection. And we found that if we used antibiotics and colonized at super high levels, that was not sufficient for a disseminated infection kind of phenotype. If you had high levels of the pathobiome and made a mouse selectively neutropenic, we were using kind of monoclonal antibody depletion, that was a plus minus. Plus, if it was something like Pseudomonas or E. coli, but not sufficient for Candida albicans. So we could colonize a mouse with Candida albicans, make them neutropenic for up to 21 days with ANCs of less than 100, and still no disseminated disease, right? If we had high levels of bug, but damaged the gut, we were just using dextrate sulfate sodium, DSS, that a lot of folks use in kind of colitis models, again, not sufficient. But if the perfect set of bad things happened, all of them, neutropenic, gut damage, and high levels, that was the perfect bad storm. And that was very reproducible every time, like 100% disseminated disease, very reproducible. What was really interesting is we found that if and what I'll talk to you about here too, is it seemed like we could only reach a kind of potential like maximum ceiling level of Candida colonization. It was 10 to the 8th was the maximum we could get in either an antibiotic treated or a germ-free mouse, whereas commensal bacteria can reach like 10 to the 11th, 10 to the 12th, this is CFU per gram. If we challenge the mice before they reach that kind of maximum ceiling, let's say if the Candida albicans levels were like 10 to the 4, 10 to the 5, and then we made them neutropenic, gut damage, it was a kind of a plus minus situation, meaning that some of the mice died, but not all of the mice died, suggesting that the level of Candida burden was indeed kind of akin to a virulence factor. So if you look at the patients that I treat now, these are the main things that seem to lead to disseminated bacterial and fungal infections. And so the question you would ask, at least that I ask as a clinician and obviously interested in translational science, is there anything I could do in the human to address these things? Isn't there a medicine I could give to make the barrier integrity of these intestinal epithelial cells better? And in fact, Amgen produced recombinant FGF7, something called palifermin. It was FDA approved, and this is kind of this landmark New England Journal study in 2004. And the reason why we don't use this is really a financial one, which is quite sad, is that the cost for using palifermin is like $10,000 a day. And if you're going to do a bone marrow transplant and then the patient's going to be at risk for many, many, many days, no one was really willing to add that price tag to the cost of a transplant, which is going to cost $200,000, $250,000 over the course of treatment anyway. So this is really not kind of pervaded or become standard of care among the transplant community. Again, I do use it in severe mucositis, but I don't use it for every single patient. And if neutrophils are so important, why don't we just transfuse neutrophils back into patients? And again, we've tried this. And this is kind of the results of a study that the NHLBI sponsored in a randomized control study looking at patients getting either granulocyte transfusions or not. And the results were equivocal. So do we do it in certain situations? Yes. Does it help sometimes? Does it do nothing sometimes? Yes. And then sometimes it makes things worse, meaning that it can kind of sequester the lungs and call kind of like a cytokine storm. So we pose the question, is there something we could do about the levels of particular pathobionts? Could we modulate that and change that? And could that actually decrease the incidence of infection? So the first kind of really, I think, compelling evidence that this is not just a mouse phenomenon or a preclinical model phenomenon was by Eric Paymer and Marcel Vandenbrink at Memorial Sloan Kettering, where Tobias resides now, in a study in which they looked at stem cell transplant patients. And if you look at patient A here, this is their microbiome. And don't get caught up in the taxonomy. That obviously this is not a microbiome talk, but in general, there's lots of colors, lots of diversity. That's kind of a healthy microbiome. But as you can see over the course of the transplant, and day 0 is when they get the stem cells, you can see that the patient A's microbiome completely transforms into being dominated by the color green. Green is what? Enterococcus. And this patient got what? Vancomycin-resistant Enterococcus bloodstream infection, as did patient B. Coincidentally or not, when they were monocolonized, essentially, or dominated by Enterococcus in the gut. In contrast to those patients that did not get dominance, as you can see here, they didn't develop. Now you could say, Andy, what about patient D? At day 35, they're essentially monodominated with Streptococcaceae or Streptococcus species. Why didn't they develop streptococcal bloodstream infections. And what's interesting is in our hands, in preclinical models, we can have mice that are monodominated in the gut with staphylococcus or streptococcus, kind of the yellow, and they don't seem to disseminate for some reason, even with high burden. But if there's enterococcus or pseudomonas or E. coli, those guys definitely translocate. So these data were really kind of striking and exciting for me. And in terms of the Candida field, Bing Zai, who was then a postdoc in Tobias's lab, they published this beautiful paper in Nature Medicine showing that indeed fungal burden, this is fungal burden in the gut, in patients who got candidemic was much higher than those who did not get candidemia, suggesting that perhaps this idea of GI burden is akin to a quote unquote virulence factor in the right particular host. So this concept of colonization resistance has really been around for a long time. And essentially what it is how does the gut, and I'm going to talk about the commensal bacteria and the mucosal immune system, how do these things kind of work together to prevent pathogenic microbes from colonizing or expanding within the gut? And the first paper that I could find was from 1957 when they were using a GI model of salmonella. And what they found is by pre-treating these mice with antibiotics, they could get disease with a 100,000-fold decrease in inoculum of salmonella, suggesting that the endogenous native microbiome was a kind of a defense protective mechanism there, right? So while this concept has been known for ages, and in terms of the fungal field, the most classic one is, you put somebody on antibiotics for sinusitis and they develop a yeast infection in their GU area, classic Candida, yeast infection. The mechanisms have been largely kind of mysterious up until, the last 15 or 20 years. So my colleague Laura Hooper at UT Southwestern, I believe, published the first kind of seminal mechanistic study about how this colonization resistance mechanism happened. And she found that specific commensal anaerobic bacteria could essentially agonize or activate toll-like receptors to induce intestinal epithelial cells to produce natural antibiotics or antimicrobial peptides, AMPs. And in case this one in particular was Reg3 gamma, which had potent effects against gram-positive bacteria like Enterococcus. And then Eric Paymer's group 2 years later said, hey, look, if we give these mice antibiotics and the commensal anaerobic bacteria go down, the Reg3 gamma goes down and the Enterococcus goes up. And in these antibiotic-treated mice, he then actually gave them flagellin, which activates or agonizes toll-like receptor 5. He could increase the Reg3 gamma, knock down the Enterococcus, and prevent disseminated disease. So when I saw these studies, I joined UT Southwestern in 2009. I got very inspired, and I asked the question, is there potentially a similar mechanism in Candida albicans? The one thing that always drove me crazy about this particular model here is that a large portion of commensal anaerobic bacteria are gram-positives. So they would be inducing a anti-gram-positive antimicrobial peptide. Wouldn't that be essentially like suicide? How would that be a good thing for commensal anaerobic bacteria? And then Andy Goodman published a paper in 2015 showing that commensal Bacteroidetes, which are gram-negatives, and commensal gram-positive firmicutes in the kind of dark blue and black are more resistant to antimicrobial peptides such as LL37 and 85 and PMB. As you can see here, they're much higher MIC as opposed to the pathobionts like E. coli, salmonella, all down here. And it was really due to structural differences in the LPS. And if they made B theta mutants that didn't have the right LPS, all of a sudden they became sensitive to the antimicrobial peptides and their MIC went down here. So there's a system by which the commensal bacteria are more resistant to the antimicrobial peptides in which they induce, which I thought was a very kind of clever process there. So what we found was an analogous kind of mechanism by which specific commensal anaerobic gut microbiota could induce a transcription factor called hypoxia-inducible factor 1 alpha, which then led intestinal epithelial cells to make an antimicrobial peptide called LL37 in humans or CRAMP in mice, and that this was effective in killing Candida albicans. And in a cell culture model in which we use human cultured colonocytes, if we gave a HIF1 alpha agonist, we could increase the amount of LL37, which could then decrease the amount of Candida within our culture system. And in mice, we found that if we gave a HIF1 alpha agonist in the drinking water, we could actually knock down Candida levels. And what was striking to us is that even though it was significant, it was 1.5 to 2 log-fold. So it wasn't like we were going from 10 to the 7th or 10 to the 8th down to 0. We were going from like 10 to the 8th to 10 to the 6th. But nonetheless, when we challenged these mice with cyclophosphamide, those again treated with the HIF1 alpha agonist and had this, the 1 to 2 log-fold lower Candida albicans, there was a 50% reduction in disseminated fatal disease, suggesting that you don't need to go from 10 to the 8th or 10 to the 6th to 0, but just knocking it down 2-3 log-fold could have a significant effect on the dissemination phenotype. Very kind of intriguing to us. And these are just data to show that you can really have this kind of, again, Candida albicans levels here among various antibiotic treatments, streptomycin, penicillin, streptomycin, clindamycin, metronidazole. And the point of this is that not all antibiotics are created equal in the effect in which they have on the microbiome and the subsequent effect on Candida colonization. And as you can see in germ-free mice without a microbiota, this again is kind of the maximum level, at least in our hands, that we can get Candida albicans to colonize. And interestingly, it's known in the microbiome field that pediatric or young mice and humans have quote unquote immature microbiota. And what you can see is you can colonize them without the use of antibiotics, but ultimately when they become mature, that the mature intact microbiota promotes Candida colonization resistance. And this is another figure from that paper back in 2015. And the purpose of this is just to show, begin sowing the seeds of intra and interspecies kind of heterogeneity. So these again are 3 different Candida albicans strains. And you can see, again, with the different kind of insults to the microbiome, water, streptomycin, penicillin, you see that there is some degree of heterogeneity in terms of how these guys colonize despite the same inbred strain of mouse, same treatment, a little bit different. And here it gets a little bit even more striking with Candida glabrata and propsilosis, whereas in the albicans strains, streptomycin was not sufficient to kind of abrogate or attenuate colonization resistance. With glabrata, at least with this glabrata strain and propsilosis, we could, right? So again, showing the importance of, it's not only important what mouse strain you have because they have different microbiomes, but what pathobiont strain you're using as well. And, is this relevant to the human condition? Again, from Tobias and Bing's paper, they showed, again, these patients here are not candidemic and their Candida levels were low, but their bacterial levels were quite robust. But those who developed candidemia, you can see some of them had quite lowish bacterial levels. And what they found, it was correlated to some degree with the use of kind of anti-anaerobic antibiotics that kill those beneficial anaerobic bacteria. And what they found, which was consistent with our paper, is that the 2 texts that we found to be really important for promoting colonization resistance were very, very low in those patients who got candidemia. So Richard Bennett and Jules Ennie and Tobias and I all kind of independently arrived at this observation that there were specific Candida albicans strains, which I'll go into, 529L and CHN1, which could colonize the mouse gut without the use of antibiotics. And so we started this experiment about 2.5 years ago where we said, hey, why don't we combine forces to see if we can find any kind of potential reason as to why this phenomenon was observed. And so this is a paper that came out at the end of 2021, and I'll highlight some key features here. So I think one strength of our study is that we, meaning Richard and Jules, Tobias and myself, were able to kind of replicate these results in different institutions, in different mice, showing again that this seemed to be a kind of reproducible phenomenon. So RI is Rhode Island, where Richard is at Brown in Providence, Rhode Island. And in C57 Black 6 or BALB/C mice from Jackson, you can see here that the 529L and the CH1 strains are much better able to colonize, whereas SC534, as has been published numerous times over the last 30 years, is really not able to sustain colonization within a mouse and gets to either the limits of detection or extruded. And again, you can see there's differences, obviously more profoundly seen in C57 Black 6 mice versus BALB/Cs. And then Tobias, again, Black 6 mice from Jackson, again, showed a comparable phenotype in which 529L and CH1 are much better colonizers. And we use C3H-HEN mice from Envigo, showing, again, a comparable phenotype. So now the big million-dollar question, which is, why? Richard did some additional experiments, which I thought were very clever, in which he did competition experiments, throwing CHN1 and SC534 at the same time in mice, and again found that CHN1 would easily outcompete SC5314 in the guts of Black 6 mice from Jackson. And he also found that true with 529L, again, in Black 6 mice from Jackson. So kind of pretty compelling that there's something different about 529L and CHN1, or perhaps there's just something different about SC5314 versus these other clinical isolates. So, obviously the question is why? One of the first things we looked at, and I'm not going to share the data because it's obvious, it's largely negative, is it did not significantly affect the composition of the bacterial microbiome. So that was not one thing that we could hang our hats on. Richard and Jules did a number of studies, transcriptomics and biolog assays, looking at the metabolic properties. They used different carbon sources. While there were differences, again, there was nothing for us to really hang our hat on. And then Richard and Jules did find a pretty striking in vitro phenotype in which they found that the 529L and CH1 had a reduced capacity to filament under various conditions compared to SC5314, with 529L really being, and this kind of picture really shows it. So 37 degrees, aerobic growth, the classic kind of filamentation assays, SCD spider and Lees plus and as you can see here, 529L, regardless of these various circumstances, is really not filamenting very well. And so we thought perhaps with 529L, we might have a putative mechanism, because as many of you know, there had been much published data that the yeast form is perhaps preferred or advantageous in the gut of mice. So again, Richard and Jules did some really nice kind of imaging experiments here. Let me just guide you through it. Candida is in the red. The green is the mucus. So you can see here, Candida here, mucus, DAPI stain here. So these are the intestinal epithelial cells. And then you can actually go see whether they're hyphae or in the yeast form, right? And in the lumen, you can do the same sort of things. And they were able to quantify this. And to our surprise, and again, showing the importance to do in vitro in accordance with in vivo work, 529L here, as you can see in the light green, this is the percent that were in the hyphal form. They were able to be in the hyphal form, and in fact, just as well as SC534, and sometimes a little bit more. So that didn't seem to be the answer as to why 529L and CH1 are better colonizers than SC5314. So taking a lead from our previous work, Animesh Mishra, who's a graduate student in my lab, wanted to see if there was maybe some inherent differences in antimicrobial peptide sensitivity, or basically were our strains more resistant to antimicrobial peptides. So we took our favorite antimicrobial peptide, cramp, bought recombinant cramp, and in an in vitro system, 37 degrees anaerobic, what you can see here is this is the growth of SC5314 in the presence of 5 micromolar of cramp versus 529L and CHN1. And in the various, even as we increase the doses of cramp, in every circumstance, 529L and CHN1 were more resistant, okay? A decent in vitro phenotype. And again, the importance of when you go in vivo, a little bit more modest phenotype, right? So again, if you look at the percent of SC5314 in a wild compared to CHN1 on the panel on the left, in a wild type mouse, as you can see, it doesn't do very well. But again, once camp knockout is the cramp knockout, so these mice don't produce cramp, SC5314 is better able to compete once cramp is removed versus CHN1. And the same situation with 529L, but it didn't seem to be sustained, because by day 18, obviously, it seemed to be kind of, obviously the phenotype was shrinking a little bit. And again, not surprising. Even in our previous work, we showed that it was not sufficient for a single host immune effector deletion, whether it was cramp or HIF1 alpha, that there are obviously multiple redundant mechanisms which obviously probably promote optimal colonization resistance. So one other thing, because the obvious question is, well, did you do sequencing and did you see? And so using already existing sequencing data with 5314 and 529L, and then we did new sequencing data with CHN1. Again, Richard and Jules leading the work on this. We did find that there were extensive genetic differences. CHN1 was clade A, 529 clade 6 versus the clade 1 that obviously everyone knows with SC5314. CHN1 and 529L seem more genetically related, closely related to each other than SC5314. All 3 isolates were euploid, and there's been data obviously that aneuploidy, like trisomy chromosome and chromosome 7 seems to have a fitness advantage in the GI tract. That didn't seem to be the case here. And these are some more granular details about the number of SNPs and indels and et cetera. And we did look at the gene that encodes the beta-1,3 exoglucanase that is supposedly targeted by LL37 in cramp, and SC5314, 529L, and CHN1. And while there were some differences, again, nothing really to hang our hats on. So I'm going to just wrap up the talk now by just making some general comments about colonization resistance. I think there is pretty compelling evidence that the magnitude of a pathobiont level in the gut, in our case, Candida of interest, is directly proportional to disseminated disease infection in mice, and is significantly associated in human patients. And again, I think this is not just true, obviously, of infections. Even if you're talking about autoimmunity, or even more the recent work like that alien prevented of the tumor microbiome, you know, if the bug is not there or there in large amounts, it's hard to affect a disease phenotype. So colonization magnitude, I think, is important. Now, having said that, the questions or the mechanisms of colonization resistance are quite, you know, obviously multifold and quite complex, right? So, but I think the main ones are, what are the Candida intrinsic factors? What are the genetic determinants which dictate why one strain can colonize or not? What are the host immune effectors, whether they be antimicrobial peptides or IgA or metabolites, and then the microbiome? I think those are the three integral factors which will dictate that, and it will take kind of really concerted efforts to get to those questions. But I do think, trying to be an optimist here, that if we can find these mechanisms and modulate Candida GI levels, that not only could we risk the risk of invasive disease, but perhaps autoimmunity and maybe even, you know, tumor oncogenesis. So again, I just want to give props to the folks here in my lab, particularly Laura Coughlin and Animesh Mishra, the graduate student who did the works on the 529L-CHN1 stuff, funding sources, and I'm happy to hand this off to our next speaker.
Yeah, thank you very much, Andrew, for this wonderful overview. I'm sure there are a lot of questions already, and I want to remind the audience to post all your questions through the Q&A function, ideally mentioning that you addressed the question to Andy Coe or our second speaker, so then we can sort it out and have the discussion in the end. And with this, I would like to hand over now to the second speaker. This is Professor Mike Bromley from Manchester. He is a professor for medical mycology, head of the famous Manchester fungal infection group. As most of you will know, he's an expert in Aspergillus fumigatus, pathobiology, also genomics. And while I guess several people on this planet can claim that they know a lot about Aspergillus, as Mike does, he is special in transferring this knowledge also to the development of new drugs. And I would say he's one of the few people on the planet who can claim that he has contributed to really invention of a new drug that hopefully likely will soon make it into the clinics. And I'm really interested in hearing what you have to say about Aspergillus resistance and new therapeutic options. Thank you for joining us, Mike.
Thank you very much, Oliver. Let me just start by trying to share my screen. Now, hopefully you can see my screen now. Yeah, thank you very much, Oliver. Yeah, I'd like to thank the organisers very much for giving me the opportunity to present our work today. And our work's been dominated pretty much by, in the last 4 years or so, by the development of a genome-wide knockout collection in the human pathogenic fungus Aspergillus fumigatus. It's been a Wellcome Trust-funded project, and in this presentation, what I'm going to do is try to give you an example of how we've taken that resource and used it to identify novel factors that are critical for both virulence in an animal model and drug susceptibility. So Aspergillus fumigatus, as many of you will already know, owes its success as a pathogen to its ability to be highly adaptive. As a saprophyte, can be found as a key recycler in most soils, and we've found it in flower beds and in soil from farmland and in lawns, and essentially wherever we look, really. And it can be found as a core part of the antimicrobial community and in compost heaps due to its ability to grow at exceptionally high temperatures. And we've cultured Aspergillus fumigatus at temperatures well in excess of 55 degrees. It really means that it has no problem in bypassing the thermal barrier imposed by potential mammalian hosts. But despite the fact that Aspergillus fumigatus spores are plentiful in the air, and we all breathe in several hundreds of these spores every day, individuals with competent immune systems rarely succumb to disease. But for those with imbalanced or poorly functioning immune systems, the fungus is able to exploit the weaknesses and invade. And it can employ a series of strategies to avoid detection, manipulate the immune system, and overcome the challenges of antifungal treatment. And it does this very successfully. There are around about 1 million cases of invasive aspergillosis each year, and these are estimates that come from Gavi, the Global Action Fund for Fungal Infections. So 1 million cases of invasive aspergillosis each year, with mortality rates around 50%. And about 3 million people suffer from chronic infections that require long-term treatment with antifungals. And a still larger number are affected by immune hyperactivities to fungi that cause allergic disease. Unfortunately, there are very few treatments available for infections caused by Aspergillus. And in essence, we're restricted to just 3 classes of compound: the azoles and amphotericin B, which target membrane stability, and the echinocandins that target the cell wall and synthesis of the cell wall. But it's the azoles that we rely most heavily on, because in terms of drugs to treat fungal infections, they're really well tolerated, well, generally well tolerated. They're highly effective, and actually they remain the only oral class of antifungal that are available to treat infections caused by Aspergillus. But unfortunately, resistance has emerged to this class, the azoles, and through work carried out by a number of labs, work we've done in Manchester and other groups in Manchester, work done in the Netherlands, and this great study from, let me just get my pointer up. So this great study from the group in Imperial, so Joe Rhodes and in Matt Fisher's group, have shown that azole resistance in invasive aspergillosis has been primarily driven by the use of azoles in agriculture and horticulture. Interestingly, in chronic forms of disease, resistance is driven primarily by long-term treatment with antifungals. And I just want to pause for a second to think about this, because this is really important. When resistance emerges during treatment in a patient, it's self-limiting. So you very rarely see transfer of resistance from one patient to another. It's self-limiting. It will, if the patient succumbs to the infection, the azole-resistant infection will die with the patient. But if resistance occurs in the environment due to use of agricultural agents being sprayed, resistant isolates can and do persist. And it's the use of azoles in agriculture and horticulture that's the biggest threat to sustainability of the azole class, ultimately for use in the clinical setting. So combination therapy is often the go-to solution for cases where antibiotic resistance is a problem. And using antifungals from 2 different classes would likely reduce the rate of resistance emergence in hosts. However, supplementing the azoles with other antifungals is rarely a first choice clinical therapy. And there are a number of reasons for this. For example, the polyene amphotericin B is really poorly tolerated in patients. And as amphotericin and the echinocandins are only available in injectable form, there are complications when treating patients at home. So it really remains the azoles that are used primarily for first therapy. But significant efforts have been made in the last few years to develop new antifungals, and this is a snapshot of the drugs that were in development or had been under development around 2015. And now fast forward to 2021, and we can see that a number of these drugs are close to making it to the clinic. And I want to highlight 2, fosmanogepix and olorifim. They are orally bioavailable antifungal agents targeting completely different mechanisms of action. So they really have the potential to become useful for the long-term treatment of patients. But if the thought is that these new antifungals will prevent the emergence of resistance and resistance won't emerge to these compounds, well, that certainly isn't the case. A little bit of unpublished work from a colleague, Mike Bottery in Manchester, has shown that resistance can emerge to olorofim just in the same way as it does to itraconazole and voriconazole. And if you have strains that are hypermutators, then the frequency of resistance to olorofim is going to be much higher. I guess that's quite obvious. But one other thing about these novel compounds is that early stage development rarely takes into account the use of antifungals in combination. And this probably isn't surprising because it's hard enough to develop an antifungal in the first place without considering how they're going to work with other agents that are currently in use. But we've shown quite recently that one of these novel antifungals, olorofim, has some synergistic effects, or at least the azoles have some synergistic effects against olorofim. And I hasten to add that although we've seen some antagonistic effect, this is relatively mild, and it's not clear whether this would have any clinical significance. As well, because olorofim is usually dosed at really high levels that far exceed the concentrations that we see here, causing antagonism. We have to take this data or use this data very carefully. So I'd like to just pause slightly and as well and highlight one potential problem that is brewing. I've already mentioned that it's the agricultural use of the azoles that's the biggest threat to sustainability of the azole class in the clinic. Well, it seems as though history might be repeating itself, because last year the US Environmental Protection Agency approved the use of a novel antifungal, agricultural antifungal called ipflufenequin. And worryingly, ipflufenequin targets the same drug as olorofim, which is this enzyme called DHODH. And there's another compound that's currently in development called aminoparaffin that inhibits an enzyme called GWT1, and that is the same target as hit by fosmanogepix. PICs. So two of our most promising new antifungals are already under threat, and they haven't even been approved yet for clinical use. So this is a concern for the future. So there are clearly drivers that mean that we shouldn't stop developing new antifungals, but can we approach the problem of drug development in a different way? Can we identify novel drug targets that will support the development of compounds that synergize with existing antifungal agents? So we've taken this question and applied it to our genome-wide knockout collection to see if we can answer the question. So as I mentioned in the opening of this talk, we've been working to develop a genome-wide non-mutant library in Aspergillus fumigatus. I don't want to go into any real detail about how we've done this. However, I want to point out that each mutant in our collection has been fitted with its own genetic barcode. And these barcodes allow us to monitor the abundance of each strain in pooled fitness experiments using a methodology termed BARSeq. Our knockout library currently includes over 6,900 isolates. So there are some strains that are represented in multiple copies in the collection, but these represent knockouts of 5,649 individual barcoded knockouts for different genes. So BARSeq, as I mentioned, has been used to great effect in bacteria and yeast to screen large numbers of strains, and it's uncovered different mechanisms of enabled uncovery of different mechanisms of drug action, and also identified novel functions for unknown genes. But for those of you who are unfamiliar with how BARSeq works, let's just take an example of a 3-member mutant collection. This collection is strain A, strain B, and strain C. Each has a different genetic barcode here, colored in green, red, and blue. These barcodes are flanked by common sequences that allow us to amplify the barcodes by PCR. So we can pool these strains in equal quantity and then extract the DNA PCR amplify the barcodes, sequence using next generation sequencing, and can use that next generation sequencing to count the frequency of each knockout in the collection in a similar way that we would apply the sequencing technology to RNA-seq data. And we can apply a selection pressure to the pool, and this would inevitably have an impact on some strains in a different way to how it would impact others, and this should be revealed through sequencing again, the pool, the barcode pool. So we've carried out these experiments for the genome-wide collection for our pool of 6,903 isolates, and we've placed them under different selective pressures. Here we're looking at selection under voriconazole and itraconazole, and these are the outcomes of our experiments. So if we're looking for targets that are that may be synergizers with antifungals, with existing antifungals such as itraconazole, we'll be looking down here. So these are the strains that have reduced fitness in the presence of itraconazole. Targets that appear on this side would essentially be antagonizers of itraconazole. We get similar profiles. So each of these, so this picture represents the all of the data from the 6,000 strains. So you can see that we got this nice distribution, some high fit and some low fit strains here. We get the same from itraconazole and voriconazole. Now we place these on an XY plot, we can see, as you may imagine, given the agents from the same antifungal class, you can see that there's a significant level of correlation between the data sets. In other words, those strains that are better adapted to the azoles all appear here, and those that are less well adapted to both itraconazole and voriconazole appear here. So these would be our antagonizers, and these would be our synergizer targets. So let's look at the bottom left quadrant now, our synergizer area, and we can see strains that we would most definitely expect. So strains like CYP51A and CYP51B. So these genes encode the protein that responsible for production of ergosterol, and this is the target for the azole class of antifungals. So not surprising that we see those here. We also see CDR1B. So CDR1B, as we've already shown, is an efflux transporter, so it pumps out azole. So loss of function of CDR1B makes Aspergillus fumigatus susceptible to voriconazole and itraconazole. And we also see some genes that are responsible for regulation of the entire sterol production pathway. Things like INSA, which encodes INSIG, a protein that couples the sterol regulatory protein in the ER. So loss of function of INSIG means that this is no longer coupled in the ER and transfers into the nucleus and activates expression of genes responsible for sterol biosynthesis. And we also see genes responsible for processing of this same sterol regulatory binding element. So loss of function of these genes means that we don't get proper processing. So all of these things we expected to see in our collection, but one enzyme that we were surprised to see in our data was an enzyme called YAC-A. Now, YAC-A is an ortholog of a protein kinase called YAC1, which has been extensively described in yeasts. However, a link to azole adaptation has not previously been described for YAC-A. As YAC-A encodes a druggable enzyme, and namely a protein kinase, and it's in this quadrant that earmarks potential targets that are azole synergizers, we wanted to understand a little bit more about what this kinase was doing. So using a subset of the library that contained only the protein kinases, we were able to replicate the fitness defect we saw for the YAC-A mutants. And we could show that in isolated culture, so this is the YAC-A mutant here, in an isolated culture, we could see that the YAC-A strain fared very badly in the presence of low levels of itraconazole. Another advantage of having the barcoded library was that we could perform pooled strain models to assess fitness in mice. So here we've pooled 25 different strains and injected them all into mice and looked to see how fit each strain is in the model. And we can see here that YAC-A, which is this orange dot here, has a modest, however detectable, decrease in virulence in our animal model, or at least fitness in our animal model from this data. And this is the data here from the individual mice. As you can see, that the fitness for YAC-A in the different replicates is somewhat variable, but differs very significantly from the control isolate, which is this one on the right. So just to reaffirm that what we were seeing for the YAC-A mutant was a decrease in virulence, we performed a standard infection model using the YAC-A mutant. We could see that, as we expected, the YAC-A mutant was attenuated for virulence. But this was not the only defect we observed for the YAC mutant. It failed to adapt to environments with low iron, with an alkaline pH, and at high temperatures. And that's despite the fact that the mutant itself had a negligible phenotype on standard culture conditions. So it's really interesting that although when we grew the YAC-A mutant in liquid cultures, we saw almost no difference between the YAC-A mutant and the wild type strain, and certainly no discernible statistically significant difference in growth between the YAC-A mutant and the wild type strain. It was clear that the mutant had a significant defect when exposed to high agarose concentrations, which seemed to suggest that it had difficulty in penetrating the media. And indeed, when we look at cross sections through agarose slices, we could confirm that this indeed was a problem for the YAC-A mutant. At 3% agarose, we can see that the wild type strain penetrated very efficiently into the media. The YAC-A was also doing okay, but perhaps a slight defect in penetration. But when we reached 4% agarose, we could see a very stark difference, with the wild type struggling a little bit to penetrate through the media, but YAC-A was unable to breach the media surface. So it seemed clear that the YAC-A mutant had a penetration defect. So the strain had quite a number of issues, and one of the ones that we were particularly interested in, because we thought it was really very relevant to the host response that we saw, was the inability of the YAC-A mutant to grow effectively in low iron levels. And while we were carrying out this study, a piece of work was published in PLOS Pathogens in Fusarium graminearum, which seemed to, on the face of it, explain what might be going on with the YAC-A mutant in respect to its inability to cope with low iron environments. The group suggested that YAC-A phosphorylated a transcription factor known as HAPX. Now, for those people who don't know about HAPX, HAPX is an iron homeostasis regulator, and HAPX is responsible for activation of genes involved in iron acquisition. So this all seemed to make sense to us, but when we looked closely at siderophore production in our YAC mutant, what we saw was no significant difference in the production of TAF-C. Now, we would really very much expect, if YAC-A was impacting HAPX function, that we would see a big difference in the strain's ability to produce siderophores, but it certainly wasn't the case. So we thought that something else might be going on here other than activation of HAPX. So what we know about YAC-A is mostly informed from yeast, and maybe I'll take the opportunity now to tell you what the data in yeast suggests. So in yeast, YAC is a stress-activated kinase under negative regulation by another protein kinase called TOR, and upon activation, some sort of stress initiates activation of YAC-A, and that's imported into the nucleus. And on import, it activates or phosphorylates a number of different transcription factors that are involved in various things from nutrient acquisition to stress response. And the evidence for import into the nucleus is pretty strong. So in Saccharomyces cerevisiae, if you take a GFP labeled YAC1 strain and expose it to stress, you can see that YAC1, which is this green color here, localizes to the nucleus as outlined by a DAPI stain here. So to uncover what YAC-A was doing in Aspergillus fumigatus, we carried out a couple of experiments in tandem. The first of these, and by far the most complicated, was to perform a comparative phosphoproteomic study. And here we received an awful lot of help from Olaf Niemeyer and Thomas Kruger from Axel Brockager's group in Jena. And we looked at the phosphoproteomic response to iron. And here you can see that those phosphopeptides with increased phosphorylation are shown in red, and those in blue with decreased phosphorylation are shown in blue. So on exposure to iron, we do see very big differences in the phosphoproteomic context. And on the YAC1, this is somewhat different. So when we look at YAC1, we can see some clear differences, and these are the YAC1 dependent phospholipeptides. So first of all, if I talk about the YAC1 independent peptides, phosphopeptides, we can see that they are enriched for genes that are quintessentially associated with iron homeostasis. So these are things like iron transporters and siderophore biosynthetic genes. For the YAC-A dependent set, however, these included proteins that are associated with cellular structures at the cell tip and the cell septum. So this leads me on very nicely to talk about the experiments where we looked at localization of YAC1. So in the absence of any stress, YAC-A seems to be dispersed throughout the cytoplasm. But when we place our GFP labeled strain in low iron environments, we see clear localization, not to the nucleus, but to the septa. But very much like in Saccharomyces cerevisiae, YAC1 or YAC-A appears to be negatively regulated by TOR. So if we treat Aspergillus strains with rapamycin, we see very clear localization to the septa. But actually, when we look closely at this, it's not just the septa, but a very specific part of the septa, and it's the septal pore. So right in the middle of the septa here. Well, what this is clearly telling us is that YAC-A has been repurposed in Aspergillus fumigatus, so it's not carrying out the same function that it is in Candida species. So what is a septal pore and what's its function? Well, septa are cross walls that enable separation of hyphae into separate compartments. The pore allows transfer of cellular materials between compartments, and upon stress, notably when a hyphal tip bursts, the septal pore is blocked. The organelle that blocks this septal pore is called the Warrening body, which is this thing here. From work in Frank Erbler's lab, we know that the Warrening body is tethered at the septa in fumigatus by an interaction with a protein called LAR. And LAR is this green protein labeled here. And this is stabilized by another protein called SPA10, or septal pore associated protein 10. If we look at our phosphoproteomics data, we can see that both SPA10 and LAR are heavily phosphorylated in response to iron, and the abundance of these phosphopeptides are significantly altered in the YAC1 mutant. So it looks as though YAC-A may be regulating SPA10 and LAR. So next we looked at the impact of loss of YAC-A on the localization of some of these septal pore components. So I'm just going to show you the LAR LAR localization for the moment. So under stress activating conditions, LAR localizes to the septal pore in the same way that YAC-A does. If we take a line across here and plot the intensity of the GFP signal coming from LAR-C, we can develop a plot that looks like this, that shows that you're getting a very acute peak right in the middle of the hyphae. And the hyphae are delineated here by these dotted lines. So a peak right in the middle. So now when we knock out YAC-A in this background, what we see is that localization of LAR-C completely disappears. So we don't see that intense peak right in the middle in response to iron stress. So it's clear to us from this data, and actually data which also shows that other components of the warning bodies, such as Hex-A, don't localize appropriately in the absence of YAC-A, that YAC-A is regulating this septal blocking. Okay, so this study really is about drug discovery and drug development. So the key question here is, can we target YAC-A to prevent this sort of localization and to synergize with the azoles? So this is some data generated using AlphaFold, and this is an AlphaFold structure for YAC-A, and we've done some docking of compounds with this YAC-A structure, and we can see that compounds are docking at 3 specific points within the YAC-A protein, delineated by these things: P1, pocket 1, pocket 2, and pocket 3. So pocket 1 happens to be the active site of the kinase. So it seems as though drugs can dock into this molecule, but the question is, can we develop a compound? Well, fortunately for us, and very much serendipitously, at the same time that we were carrying out this work, there was a publication by Jesse McAlpine in Leah Cohen's group that described a small molecule called ECBC, which was an inhibitor of YAC1 in Candida albicans. And it appears that YAC1 docks really nicely in this structure. So we're really happy to see this. So we tested this compound against Aspergillus fumigatus. So we can see here, so this is what happens to YAC-A localization in the presence of low iron. So as we introduce a low iron environment, you can see a buildup of YAC-A at the septal pore, very much as we'd expect, and we get very high signal after 60 minutes. But now what happens when we add this compound ECBC, we can see that we no longer get accumulation of YAC-A at the septal pore. Now that's really useful, and it very much suggests that YAC-A might, the ECBC might be inhibiting YAC-A. But the key question, and what we really started with, was, can we identify synergizers with the azoles? And actually now, if we do a checkerboard assay using ECBC and the azole voriconazole, we can see very clearly that there is a wonderful synergy between these molecules, very much as we expected. So just to summarize then, I think, I hope I've convinced you that competitive fitness profiling is really powerful, and it can be exploited to uncover mechanisms of adaptation. In Aspergillus fumigatus, YAC-A has been repurposed to regulate the plugging of septal pores in a TOR-C-dependent manner, and we feel as though YAC-A is a promising target for the development of novel antifungals, and that it just leaves me to thank some of the key players in this process. I really want to highlight the guys that have been involved in the knockout library. So Paul Bowyer and Daniela Del Neri have helped to lead this project, but a lot of the knockout work has been done by San, Lauren, Marcin, Izzy, and Risa. And I'd like to thank very much Axel, who has also been very supportive, and his team in the development of the knockout library. And I think I'll leave it there. Thank you.
That was fantastic. So now we are open for the Q&A. Again, we remind everybody that they can put their question in the Q&A chat. So I will start with the first question for Andrew Ko. You mentioned that the strains that seem more like commensals are more resistant to CAMP than SC5314. Is that also true for other species of fungi that colonize but have lower virulence than Candida albicans? For example, Candida glabrata, does it have lower susceptibility to cramp?
Yeah, that's a good question. I think the challenge here is that it'd be nice to do some sort of comprehensive screen to find the major AMPs or metabolites or immunoglobulins which really modulate these things, and then be able to screen specific strains to see their susceptibility to these. The LL37 thing, I don't want to oversell it. It's a proof of concept, right? But I think those would be interesting. We already have some preliminary data that we've generated in kind of unbiased screening approaches where we identified metabolites that are negatively associated with Candida colonization resistance. And we find that the strains that we just mentioned are also differentially susceptible to the inhibitory effects of those metabolites as well. I think it's like this kind of cumulative effect of multiple immune effectors, which will dictate whether it can do it or not. So, sorry, long-winded answer, but no, we haven't done it, but that's something I think that needs to be done, sure.
All right, thanks.
Right, so I'll continue with questions for Mike. And again, there's first a lot of compliments for your talk, clearly. And I'll start with a question regarding the unequal fitness of the CYP51B mutant, if you compare ITRA and VORI. Do you have a reason why you found this? Can you explain this? You're still muted, sorry, Mike.
Yeah, thanks, Oliver. Yeah, that an absolutely fabulous question, and it didn't escape our attention that while CYP51A seemed to be equally unfit in both VORI and ITRA, CYP51B was much less fit in VORI challenge compared to ITRA, and it probably is something to do with the differential ability of each compound to dock and inhibit CYP51B versus CYP51A. We haven't carried out any experiments to directly prove that, but I suspect that will be the case, that it's something to do with itraconazole, which is rather a large molecule, being able to access CYP51B as well as voriconazole.
Right, and then I have another question here, and this is about the phenotype with the penetration into the agar, and the question is if you can compare the forces needed to penetrate into 4% or 3% agar with forces that you wouldn't need in the in vivo situation, and then a more general question of whether we all need to have this as a screening assay in future characterization of mutants.
Okay, let me deal with the question about whether or not it's really physiologically relevant, and the answer to that is I don't know, and it's something that we really need to look at. Unlike an agarose gel or an agar gel, the lung is not an homogeneous entity, so to penetrate through various parts of the tissue or certain membranes, or there are going to be different forces required to penetrate through different parts of that lung, and I don't know at the moment what strength of pressure would be needed to penetrate through. So is it physiologically relevant? I don't know. What we do see in our animal model is that when we try and when we do histology, we don't see that the YAC-A mutant is able to penetrate through through the alveoli into the surrounding lung tissue. So it may be the case that it could be physiologically relevant, but that YAC mutant does have a number of other phenotypes that may be stopping it from penetrating as well.
Okay, I know that there are some studies about the forces required for Candida hyphae to penetrate cells, and I'm somehow I forgot the name. I'm sure I can think of it during this talk, and I'll send you something because I also think that's an interesting question. Another question here is about the role of Candida albicans YAC1 in adaptation to hypoxia, and whether this is conserved in Aspergillus.
So we do see a hypoxia-associated phenotype with the YAC1 mutant. So it does grow less well in low oxygen environments. However, we haven't assessed that in any great detail. So we don't know exactly what's going on in hypoxia and whether or not the YAC-A GFP will be localizing to the septal pore, or if indeed it's going somewhere else when placed under different stresses.
All right. So next question for Andy Koch. So you mentioned that anaerobic bacteria from the gut or Klebsiella as well, their Enterococcus, in addition to Candida, are translocating and causing those infections. So why staph is not doing that? Is that, or Streptococcus, is that something to do with the site from there they infect? Or is that something to do with their biology? Do you have some ideas?
Yeah, I mean, I'm just going to speculate. I think, the taxa that are there, is it like Staph aureus or Streptococcus pyogenes, which are known to have virulence factors that in the nasal cavity can allow invasion. It seems like they're distinct Staph and Strep taxa. And again, my hand-waving answer is I think they're just not capable of so if you take Pseudomonas and obviously remove their virulence factors, they can also, not, despite the fact that they're abundant or et cetera, won't be able to disseminate. So I think they're just less virulent Staph and Strep that are in your gut that don't allow them to disseminate. Yeah.
Thanks.
Yeah, Andrew, can I also ask a question? So that's unexpected because it's not in the chat, it's from me. I was wondering, these strains which have an improved capacity of colonizing the gut, do they have a reduced or even elevated capacity to cause disseminated infections? Or do you see a strain?
That's a great question. So we haven't done the experiment now where we challenge them with, chemo and et cetera. Those are ongoing and we'll see whether they, great question.
Okay, so then I got reminded about the authors of the forces needed for hyphae in Candida, and I'm really sorry because I should have known. It's Alex Brandt from Exeter and Rob Arkowitz from Nice, and here are the names. And then there is another question, and this is again about CYP51A versus CYP51B. And the question, if you can speculate, Mike, about what this means for other fungi, for example, haploid Candida. How can you transfer your findings to perhaps other pathogenic fungi?
Yeah, so gosh, difficult question. So I think in Aspergillus fumigatus we have two CYP51 genes, A and B, right? And there's a certain level of redundancy there. If we knock out either one, the cell remains viable, but if we knock out both, the cell won't survive anymore. So aldosterone biosynthesis is critical. There are definitely other things that we can target in the aldosterone biosynthetic pathway in both yeast and in Aspergillus. HMG1 would be a prime example. So there are already compounds out there, such as the statins that target HMG1. And they are active against Aspergillus. Whether they're active against Candida, I'm not sure. But they certainly could be used as an alternative target for the same pathway.
Okay.
Okay, so another question for Andy Koch. So the beta-1,3 exoglucanase is targeted by CRAMP1, but you said that there are some variations. Do you think actually that the strains are able to show that differently? Is it some structural problem between strains, or what do you think it is?
Yeah, again, I want to apologize. I'm not I can't turn on my camera for some reason, so I'm not avoiding doing this. Well, you know, Ilian, that brings up a really interesting question, because I think I alluded to that one of the reasons the commensal, like Bacteroides and Firmicutes, can resist the antimicrobial peptides is that their LPS structure is somewhat different. So maybe, you know, it seems like the genetic differences aren't different, but they could lead to structural differences. And again, it's something on our plate to do. Part of the challenge is my research program has really grown over the last 4 years, but everybody wants to work on the cancer microbiome stuff. So I have to enlist some folks to work on the Candida stuff. So, but I think that's a great question. Yeah.
Yeah, thanks. I hope with the MycoTalks more people get attracted to work on fungal pathogens. It's important, and who is at the conference already knows it, but I hope we broadcast that on the internet with those talks, and yeah. So.
Okay, Mike, I have another question, and this is with your idea of looking at the synergistic effects of new substances with known antifungals. Do you think that this is a good way of just finding new targets and new drugs, or would you really think that we should consider going towards combination therapy of different drugs at the same time?
So, I mean, it's really hard to develop an antifungal, exceedingly difficult. You can just see by how few antifungals there are available and how long it has taken to develop these new compounds like olorofim, fosmanogepix, razorfungi, and all of those. It's going to be, so there are a lot of economic drivers that mean that it's quite hard to develop a compound just as a combination therapy, because it's much more difficult to do the human models, the clinical trials. You're limiting yourself in terms of the number of compounds at the very early stage that you're looking at, and the number of targets. I think what we possibly can do is look a lot more at the chemical space that there is available, and use those in combination with existing antifungals to try and find synergistic molecules. And I know that a number of groups have been trying to do this, and it seems relatively sensible, including, you know, drug libraries of already authorized or approved compounds. So that seems sensible to me. But whether or not there's going to be the sufficient financial drivers to allow those very expensive clinical trials to be carried out, I just don't know.
Okay, so then thank you very much from all of us to both the speakers and everyone who contributed with questions. Not as many questions as we are used to in the MycoTalks series, probably because a lot of people are in the session at the GRC conference.
Yeah, it's ongoing.
Yeah, but this also means that we will likely break the records for online on-demand viewing because of the quality of the two talks. So it has been a pleasure to share this with Ilian. I want to remind everyone that the next MycoTalks will be 23rd of February, and we are all looking forward to seeing you again there. Thank you very much.
Thank you.
Thanks to the speakers, and thanks, Oliver. That was a pleasure.